While it's being set up, let me thank the organizers for the invitation, Anupam, Ganga, and Raghu, and also congratulate Maha. So while it's being set up, let me tell you a story. So I first, yeah, that should go. I first encountered Maha almost 20 years ago to the month. This was a meeting in ICTP, driven states in soft and biological matter that Madan, Sriram, Gautam, and Jack had organized. And there was a buzz about that Maha is coming the second week. And so he came and he spoke about soft hydraulics, physics and physiology. I was just an MSc student. Most of it went over my head. I didn't get what he was talking about. But the thing that impressed me the most was the kind of questions he would ask. So one of the examples he gave was, we were in ICTP, curd was something that we were all craving. So he said, I have a piece of curd and let me put a spoon there and the spoon sinks and think about how the stresses propagate and so on. So that was really interesting. Damn, this is the kind of questions that we should ask there. So Maha has been somebody whose every paper makes you like, why didn't you think about it? So thanks, Maha, for teaching us how to think about the natural world. So I'll tell you about a problem which came out last year. We're fighting somewhere about this. We're trying to understand how cytokinesis, particularly cell division, can come up very naturally from differential geometry and active mechanics. So this is classic picture. Can I dim the lights if possible? This is a classic picture of cell division that we all see. Cells divide by duplicating the genetic material, then they're pulled apart and so on. But there's also this large-scale actin structure that comes up, the ring that forms, which cleaves the cell, mother cell, into two daughter cells. Classic picture that we all know. But the cell division problem is also a problem where there's dynamical geometry going on. The mother cell, goes into a peanut-like shape, and then the peanut-like shape eventually cleaves into the two daughter cells. So I want to understand the dynamics of the geometry coming up in this problem. So here's the conceptual picture that we learn from any standard textbook in cell biology, that there are large-scale flows on the surface of the cell, this structure called the actin-myosin cortex. Somehow these set up a pattern of a ring-like structure of actin-myosin. The constriction of the ring is what leads to the furrow formation and therefore the eventual division of the daughter cells. Classic results from the 80s is where we learned the cortex is the thing that causes these structures. So I want to ask if you have this feedback between geometrodynamics, that is the dynamics of the surface itself, and the patterns that come up on the surface, actin-myosin patterns, and the concomitant flows and cell shape. Can this spontaneously lead to the emergence of cell division-like patterns? Cytokinesis is what I call. And the answer is yes, and I want to tell you that story here. Okay, so first the physics of the cortex. The cortex is itself a very thin meshwork of filaments just beneath the plasma membrane. Lots of gooey material, transmembrane proteins, and so on and so forth. More important thing is that it's an active material. There's continuous supply of ATP, which myosin motors eat up, and then they tug filaments, and so they create force dipoles. A coarse-grained description of that is an active stress. The actin-myosin cortex is implicated in many cellular processes, even at the cellular and even at the morphogenetic level. In particular, if you have stress gradients which are tangential to the surface of a cell or a tissue, that can lead to flow. And if you have normal forces, that can, of course, lead to shape changes coming up there. Even at the larger scale level, the cortex is something which is implied very heavily. So one way to think about the cortex is to look at the size. So roughly, it's about half a micron in size, depending on the system, 300 to 500 nanometers. And so you can integrate out the thickness because the cell itself is several tenths of microns in size and get an effective two-dimensional description. So think about the mass density of the material in the cortex. So what I can write is an evolution equation for that, where I have a non-zero divergence coming because stuff can go in the third dimension, but there's also the third turnover. And this turnover timescale, typically coming from polymerization, depolymerization, is typically of the order of few tens to 20s of seconds for typical cells. So I can look at that dynamics and write down a force balance description, low Reynolds number. I can forget inertia. I balance forces, whatever external forces there are. I write down a stress, which is a fluid stress and an active stress. And I can write down a fluid stress in the limit where the Maxwell relaxation timescale is roughly of the few seconds. If I look at timescales much longer than that, a fluid-like description is a pretty good approximation to this problem. So once you have that, what one does is to try to model where this activity comes from, the active stresses. And so that's the theory of active hydrodynamics that people have been building from the past 20 years. So one thinks about force balance, force and torque balance. transport of density, of concentration fields, and so on and so forth. And typically, these materials also have orientational degrees of freedom, so maybe polar or nematic liquid crystals. The more important thing is that the non-equilibrium aspect, the ATP consumption, breaks time reversal invariance and leads to the emergence of generic active stresses. The active stresses go and feed back into the force balance, and also certain new terms that are allowed just by symmetry come up in the picture. So I'm going to do the following. I'm going to assume that the density relaxes faster. So I'll forget the total density. So it's more or less uniform at some steady value. And assume that since the cortex is in plane isotropic, again, the time scales for orientation relaxation is small. And so I look at an active thin film limit. And so essentially I get a force balance equation coupled to a concentration field, say a single concentration field. The concentration field generates active stresses in the plane of the cortex. And those active stress gradients drive flow, and the flow affects the concentration. So there's a nice feedback between concentration and flow. So such systems give rise to a very useful set of predictions. So one of the things that's been done is to test this idea in a real cell and to see that indeed you actually get this picture valid in a quantitative scale. So this is the first cell division of C. elegans zygote. If you follow the picture here, this is myosin that has been labeled, and you're flowing from the right to the left. You can do some particle imaging velocimetry and get a flow profile out. You can look at the flow profile, you write down this theory in one dimension, and ask if the predictions of the theory pan out with the experiment, and they do. So it gives you good confidence that this hydrodynamic picture at the level of the cell, at the level of the cortex, is something that works. It also gives rise to a new class of patterns, active patterns, which one we call. So let's say you have a concentration C, which moves around by advection diffusion and simple turnover. The C is special because it generates isotropic active stresses. Plug them back in here, gives you a flow equation. So it's coupled dynamics of flow and concentration, and you can show that beyond a certain activity strength, the homogeneous state becomes unstable and you get spontaneous pattern formation coming up. So these are active mechanochemical patterns. You should think about them as analogs, mechanochemical analogs of classic Turing patterns that you had them. An interesting thing to note is that if you don't have this coupling between mechanics, there are no patterns. The homogeneous state is the only stable steady state. So we've been exploring this in various settings, in various contexts, and I want to think about this in the context of a cell division today. So when you think about cell division, turns out that these kinds of flows are useful even in trying to understand axis establishment in creatures. Again, in the C. elegans zygote, the very first cell division turns out to be an asymmetric cell division. The two daughter cells are different in both size and also in chemical composition. The chemical composition asymmetry is set up by certain polarity patterns on the surface of the cell prior to the cell division. So these are classic PAR proteins that we know in cell polarity across the animal kingdom. And so what we did some years ago was to write down a theory for the cell polarity problem, where you have the red protein, the blue protein, myosin, and so on and so forth. And the myosin is the one that generates the flows. So you can couple the flow and the pattern dynamics, measure things very, very quantitatively. Turns out that the theory and experiment match extremely very well. So it gives you confidence that this kind of a picture works in real biological settings and in a quantitative manner. So I want to understand, how does geometry come into the game here? So in particular, I want to think about cell division, and in cell division, as I remarked, that the geometry is something which is dynamical, and how do I bring in geometry into the game? So the first simplest geometry is you think about, let's say, a sphere. So I take the same cell, I put the cortex on the surface, but this time, let's say I put in a cytoplasm in the interior as well. So I have my equations for the surface density, advection, diffusion, turnover, and a surface force balance. But the cytoplasm, I just say it's a boring Stokesian fluid, and I put in the boundary conditions for the Stokesian fluid being the surface flow. I evaluate the stresses, and so therefore the traction provides a coupling between the surface and the bulk. So this you can actually solve for the sphere, and you can show that there's really only one number that matters for the game. And so there's the ratio of the surface shear viscosity to the cytoplasmic shear viscosity. That's a length scale, and that length scale compared to the radius of the sphere dictates the kind of patterns that you can get. So if that length scale is much larger than the radius of the sphere, you get a spot-like pattern, L equal to 1 instability. That's like a cell polarity pattern. But if that length scale is much smaller than the radius of the sphere, you get a L equal to 2 instability. That's like a cell division pattern. So it's kind of a building, the building blocks for cell division picture comes up there. So for some real cells, for particularly C. elegans zygote, this ratio turns out to be in the wrong regime. So this cannot be the mechanism by which typically a cell division like ring comes up there. I wanted to explore arbitrary geometries. Remember, I want to build for dynamical geometries eventually. And so one way to do this is the usual physics that we learn in any differential geometry course. You write down the differential geometry of the surface and ask, I should write down the covariant version of these equations. As any of us know, that once you do that, all kinds of difficulties crop up. You can't do even simple linear stability analysis, anything other than a sphere or a simple plane. And so you would resort to numerics. But then there are coordinate singularities, and then it becomes really messy. So one would ask if you can build the numerics directly without going into these intrinsic coordinate descriptions. The idea is that there is this nice field in mathematics called the tangential calculus formalism, where you can actually write down the surface as embedded in the ambient space, write down equations for derivatives on the surface in terms of the derivatives on the outside in the ambient space. And it gives a very, very nice picture. This also is completely consistent with the covariant picture that we did. So what we did was to develop a formalism where we can solve for such patterns on any arbitrary surface, even arbitrary topologies. So what do we see? So one thing that we see very clearly is that the geometry is very, very crucial in determining the kind of patterns in active mechanics. So the same model that I showed you earlier, if you put that on a curved surface, just because of the calculus, the Gaussian curvature comes up explicitly, but there's also curvature buried in the derivatives and so on. So the kind of patterns that you see, they seem to be very sensitive to the geometry. So sometimes you see spot-like patterns seeking high curvature or low curvature, spontaneously breaking symmetry, and so on and so forth. Okay, so I want to come back to cell division. So I asked, how do we see that this geometric feedback and between myosin and geometry, and can that lead to cell division spontaneously? So this is work done by my student here at ICTS, Aditya. He's moved on to Francis Crick Institute a couple of months ago, and Masayoshi Nishikawa from Hosei University, who did all the experiments there. So I showed you a bunch of patterns where you think about, say, pattern formation in a plane, or maybe on a sphere, or maybe on spheroids, and so on and so forth. This is a problem where the geometry is fixed, right? So, but the cell division problem is a problem where the geometry itself is dynamical. And the dynamics of the geometry is not something where you impose the geometry dynamics, but the geometry dynamics comes from equations which are on the geometry, right? So there are fields on the surface which dictate how the geometry changes, which dictates how so there's this feedback that you really need to build. And so is it 5 minutes? No. Okay. So the way you think about patterns and dynamical shape and the coupling between patterns and dynamical shape, and this is what I call geometrodynamics. For people who have seen geometrodynamics in other contexts, it's similar to that aspect. So here's the conceptual picture. I want to start from something which is more or less unpatterned, homogeneous in some concentration field. The hope is that you'll get patterns, and the patterns will change the shape will change the patterns, and so on and so forth. I want to ask, what are the kinds of emergent patterns that come up in this case? Okay. So again, if you want to do this for a dynamical system, you again have to extend this formalism typically by writing down equations of motion for the metric tensor and the curvature tensor and so on. Again, one can do this, but can calculate only for very, very simple things. And so in fact, many people say everybody writes down these equations, but nobody solves them because you can't solve them. So what we did was to extend the formalism that we did for the tangential calculus to dynamical surfaces. And so you take a surface, you can look at the velocity, has normal components, tangential components, and so on and so forth. You can show very easily that the curvature is related to the gradient of the normal. And so if you say this is a fluid surface, where the material points move only along the normal direction, local normal direction, you can compute how the normal changes itself. It essentially changes by the gradients of the normal component of the velocity. This is just pure kinematics of a fluid surface and nothing more than that, right? So you would like to ask, where does the flow come from? Well, it will come from the forces on the surface. So before going to the active mechanics, we made sure that everything is fine. We looked at passive surfaces, put in a standard bending elasticity, some Kahn-Melfish Hamiltonian. Turns out that you can rewrite that in the language of a stress tensor. Kapoor-Laguven showed us that some time ago. And once you put in that, you have a fluid and a bending stress, and the same mechanism that we wrote down earlier. We've rechecked all of things that we know in classical membrane relaxation dynamics, spheres coming up as a final time limit, the way the convergence happens, everything goes through. So that means the numerics and the way we are putting in dynamical surfaces is correct. So now the only game is to put in the activity. Okay. So here's the full model. So we take the dynamics of myosin on the surface. Myosin generates active stresses. In addition to active stresses, there are fluid and bending stresses. And the force and torque balance tells you that the divergence of the stresses must be balanced by some frictional forces and some pressure difference. I put in a pressure difference to maintain the total volume. This is reasonably well known and well measured in experiments. So once you do that, this gives you the dynamics of the field on the surface. But then the velocity that comes up from solving the force balance will move the points on the surface, which will also change the geometry of the surface. So there's a nice coupling between patterns and geometry naturally coming up here. I would like to ask what happens as you increase the activity. So the only two relevant numbers are the Peclet number, which is a measure of how active the system is, and the Damkohler number, which measures turnover rate to diffusion. So one thing that we got, which was very nice, is that if you start out by something which is a spherical surface with more or less a homogeneous distribution of myosin, we run the system for a long time, we got the emergence of cell division spontaneously. Not just the emergence of rings, but also the emergence of a shape transition coming up very, very spontaneously. So this is the movie that you're seeing here is a sequence of time snapshots of the thing. This is completely self-organized, nothing put in by hand. And this automatically comes up in a large region of parameter space that we're looking at. Okay, so here's some cross-sectional views. So you can look at the process in the transverse plane or in the division plane, and you actually see the very nice ring-like structures coming up in both the planes, that we can actually see in the experiments. So here's the phase diagram. So in the Peclet and Damkohler space, there's the boring homogeneous state where you only have a sphere with uniform concentration of myosin. Yes, ma'am. Previous slide? Yeah. Yes, I will come back to that. Yes, yes, yes. I know you're interested in singularities. I'll come to that in a minute. Yeah. So you have the boring homogeneous state. Beyond this, which is where you can do a linear stability analysis, and you get back the L equal to 1 kind of a shape. That's something that we get in the numerics as well. Far beyond this, in the highly nonlinear regime, is where you start getting these constricting rings. So like Maha was asking, this is actually not a steady state. So it eventually becomes sharper and sharper. There's a positive feedback, the curvature increases. At some point, our numerics breaks down. So we can't go that far apart. In fact, it becomes so much that the surface starts touching each other. We can't go that far apart. So therefore, we can't go to the singularity, we can't address that. However, we want to ask, what about the kinetics of the ingression? Can I ask the question there? Yeah, so you will see some symmetry aspects coming up. I'll show you why you need the full 3D thing, because I'll address something which is not symmetric. So this is a regime where you don't have stable steady states, but you have constricting rings, and you can vary the other parameters, the Peclet number and viscosity, Peclet number and some bending elasticity and so on and so forth. In all the cases, there is a large enough region where you get these constricting rings. So, but this is not a steady state. However, can I compare the kinetics? So this is where I don't know if it's visible, if you can switch off the lights a little bit. So this is where we collaborated with Mastoshi. So he actually measured the dynamics of the ring constriction in the coeliac zygote. So this is about 50 microns by 30 micron cell, big one. So we label myosin, do confocal imaging, reconstruct in 3D, and you can actually see the formation of the ring and the constriction of the ring. So these are the two division plane and the transverse plane views of the same movie there. So we have very quantitative data in space, in time, and over many, many repeats of the same experiment. So what this allows us to do is now to go back and ask questions about the kinetics process. So one thing that came out very nicely is the following. So we asked in the theory, how does the radius change as a function of time? So I start with some initial radius, and as a function of time, I look at the radius of the ring, and I ask how does that change with time? So our theory said that there is some initial transient, and after that the radius decreases linearly with time. There is a regime. So this is something we didn't put in by hand. It just comes out of the active mechanics and the differential geometry. To our surprise, this was something that people had seen across cells, many, many cells. So in amoeba, in sea urchins, 4 different species, in C. elegans, and so on and so forth. Many people had seen this. Another thing that our theory predicted is that the ingression rate should go up with activity, with the Peclet number. So can we test this in experiments? So it turns out we can. So for C. elegans, we know a very good library of perturbations where you can knock off this gene and that gene, and that affects the properties of the cortex. For example, there are gene perturbations which will speed up the flows or slow down the flows and so on and so forth. So we did these experiments, and lo and behold, as predicted by our theory, if you reduce the flows, the slope of this straight line goes down. If you increase the flows compared to the wild type, the slope indeed goes up. Okay, so that's about the ring ingression. Yep. Coming, I'll write a theory for that. So we looked at the myosin dynamics in the ring. So we look at the ring, and you average the myosin in the ring and ask how does that myosin change as a function of time. Our theory predicted that increases exponentially with time. Okay, and in fact, in the experiment 2, we found that across perturbations, there is a clear exponential increase of myosin there. So there's one more statement that the rate constant for the exponential increase, Km, this increases with activity, and that's in fact exactly what we see in the experiment as well. So coming to Maha's question. So I told you two things. One is that the radius goes down linearly with time. The other is that the myosin goes up exponentially with time. Are the two related to each other? And the answer is yes. And it turns out that if you look at the Gaussian curvature, so you will see in the theory there is some region where the Gaussian curvature crosses 0. So that's the point where the surface becomes almost flat. So around that time point, we can actually take our equations, simplify them enormously, and write down an effective equations for the radius and the average myosin concentration in the ring. Exactly. And in fact, they show that in this approximate description, that the rate at which the radius decreases is directly proportional to the activity and some friction and some radius setting up there. And the exponential increase constant for myosin is also some combination, but we predict that U-perp and Km should be linearly related to each other. Okay, so this is just from the simplified analytical calculation that we can. Does this work out? First thing, in the theory, what we did was to vary a bunch of parameters, viscosity, Darm-Kohler number, activity, and so on and so forth. And at each parameter value, we also did various random initial perturbations to check if the dependence on the initial conditions. And as predicted by the analytical theory, the Km and the U-perp are indeed linearly related to each other. To our surprise, we asked if this works in experiments, and in fact, across 9 different perturbations of the embryo, the rate constants Km and U-perp indeed turn out to be linearly proportional to each other. Okay, so the theory of active deformable surfaces seems to be something that can capture the ingression and the myosin kinetics in the cytokinetic ring pretty quantitatively. Say it again. You shouldn't worry about the intercepts. Definitely no. This is a very approximate theory, so you shouldn't worry about that at all. One last thing to show why the full 3D thing is required. Why can't you just do assuming azimuthal symmetry? So I talked to you about asymmetric cell division, like here, but there's another asymmetry which I want to show you about, which is rather interesting. So this is asymmetry in the ingression itself. So again, the classic 2 daughter cells doing asymmetric division, but if you label the surface, say some myosin membrane marker, and follow the dynamics in time, you see something rather peculiar. One side ingresses much earlier and much faster compared to the other side. Okay, so if you look at the division plane, you see the cytokinetic ring, and the center of the cytokinetic ring does not stay where it started out with. So it actually slides there. So this is time sequence of snapshots of the cytokinetic ring that you see here. So maybe it's some peculiarity of C. ligands. I mean, living things are weird. Turns out no. Rappaport's book actually lists a huge range of creatures which actually show asymmetric cell division. In fact, here's a picture of a comb jelly where you see ingression only from one side, the other side does not ingress at all. You see something similar in the sea star as well, but there is a localization of the spindle coming up there. So we also checked to make sure that this is not some, some signal-driven thing. Whenever we do experiments, it looks like the asymmetric ingression point is random as far as we can make out. So it doesn't seem to be driven by signals. It also doesn't seem to depend on the fact that you put the cell between the glass plates and squished it. You can put it along gravity, and in fact, you still get asymmetric ingression. The cross sections are much nicer and circular. As far as we can make out, the spindle is more or less at the center throughout the process of ingression. So that doesn't seem to be the reason for the ingression. So in fact, we actually measured the spindle and the surface dynamically in time. And throughout the process of ingression, the spindle doesn't seem to be localized to either of the surfaces. So how do we get asymmetric ingression coming up? All we had to do was the following. So I showed you some symmetric ingression. You can see the 2 ingressing sites are similar in the ingression amount. All we had to do was to just increase the contractility. So as soon as we increase the contractility, we found that spontaneously you start getting asymmetric ingression. Okay, so I'm only showing you this is the cross-sectional views of this 3-dimensional thing, and you actually see the asymmetric ingression coming up very, very nicely here. This side ingresses less or more compared to the others. So we can again measure things very quantitatively. So we can measure not just the radius, but also the shift in the center as a function of time. Our theory predicts that the shift in the center should increase exponentially with time. Lo and behold, the theory matches very well with the experiments. So again, one more question like I asked earlier, is there a relationship between the asymmetry rate and the ingression rate? Our theory also predicts something which is linear. Same thing is panned out by the experiments as well. Okay, so what we seem to suggest is that the theory of active deformable surface, the full thing, captures even the asymmetric ingression part of cell division coming up there. Last thing, we also noticed something rather peculiar. So I've been talking about the cytokinesis aspects. What about the cell division aspects, the two daughter cells being said? In the real cell, there are much more complicated chemical reaction networks which sense cell size and so on and so forth. But we found out to our surprise that the turnover rate actually controls the eventual two daughter cell sizes. So if you have zero turnover, you seem to get more or less all the time asymmetric ingression. As soon as you have some non-zero turnover, it symmetrizes and the two daughter cells become similar in size. Okay, so that's the picture that I'm trying to paint, that there is a coupling between flows and geometry, flows and patterns that people have been exploring. Geometry as a dynamical variable will be really nice to explore, and there'll be plenty of things to ask questions about. Here's one thing I'll end with. One question that I always get, isn't that the spindle that's doing the job for cell division? Turns out this is a very nice experiment by Hiramoto in the 60s. So where he does the following experiment, takes sea urchin eggs, and when they're just about to divide, he puts in a pipette, can you see here? And at some point of time, he sucks out the spindle. And then asks what happens to the cell surface without the spindle. It goes through cell division. So it divides. So it looks like the cortex is a self-organized structure, can actually lead to an ingression and the eventual cytokinesis there. Surely this is one problem where there's dynamical geometry. There are plenty of problems. So we have a nice computational tool where we can actually explore many of these things in the context of tissues and so on and so forth. So I'll stop there. So what we tried to do is to see if active geometrodynamics can predict cytokinesis. We find that the self-organized dynamics of the regulators and the geometry can indeed lead to this, and it matches very well with experiments. I'll stop there. Thank you.
Thank you, Vijay. Nice presentation. Yes, so questions for Vijay.
I don't know, is this working? Okay. There are some very beautiful experiments, not with C. elegans, but with yeast, for example, which I think were done by Mohan Balasubramanian and colleagues. And I thought in some of them, you see the equivalent of an actin-myosin ring. I can't quite remember whether it's the same. Sometimes it slips off and sometimes it essentially forms.
Yes.
Can you comment on that? And maybe a second part of that question is, you mentioned these last observations from 56 from Hiramoto and company. Question over there is, do you need to go past some critical size that the spindle is needed for, after which you don't need the spindle?
Correct. So maybe the second question first. So there are some signals that come from the spindle which will initiate the process. But once you do that, you don't need the spindle. So in some sense, the critical size for ingression must be something that is coming from the spindle. So the first question that you asked about the slipping of the ring, and that indeed is something that we see in, where is the phase diagram? Yeah, here. So it turns out that here is where you get spot-like patterns, right? So this is, and here's where you get the ring-like patterns there. So in fact, close to this, you start forming a ring, but the ring slips and becomes the spot. So we mostly explored spherical geometries, but we can now make it like a yeast thing, put a cylinder or something like that. Then you will actually get a ring moving. I think that Madan has also seen in some of his earlier studies.
The same paper.
Quick question, Vijay. So the fact that you introduce asymmetry in the ingression by
it comes out spontaneously.
Changing the contractility, right? But are there other choices that would also result in?
We haven't found, because without contractility, everything seems to be boring and dead. No, if you make contractility zero, there's nothing. It's a minimalistic system. Surely the contractility and combinations of other parameters, the viscosity, bending, elasticity, that will also control that. But you definitely need to be high enough in contractility. Otherwise, you don't get asymmetric.
Okay, thanks.
Okay.
Questions for Vijay? No, so Vijay, one thing, like you did not consider stretching at all, like elastic. Stretching.
I've put in a fluid, like I have membrane elasticity, but not about the stretching of the surface itself, not the tangential stretching.
So that
I have out of plane elasticity.
Yeah, bending is
that is there.
Yes,
that's in fact there.
So, but should that play a role?
It does, because if you surely make very high bending elasticity, you cannot deform the material so much, right?
Right, but in plane stretching, like
In plane stretching, because the membrane is a fluid membrane, so that elasticity is not important. We have a surface tension. That I have.
thank you. Okay, so we thank Vijay
Thank you.
for very nice talk.